And finally, we get to stage 3, which is the cellular response. So we have allowed the signaling molecule to bind to the receptor, stage 1. We've gone through a signal transduction pathway, stage 2, and finally we are ready to actually enact some sort of cellular response, also called an output response. Typically, this is the regulation of one or more cellular activities, and the response can occur either in the cytoplasm or actually even in the nucleus. If the response occurs in the cytoplasm, this is, for example, very commonly the regulation of certain enzymes. Maybe this is going to be a change in metabolism of the cell. If the response happens in the nucleus, again, most typically, this is going to be regulation of gene expression. So you're going to be turning on genes, turning off genes, all dependent on what the cell requires. So if we take a look at this example here, this is looking at how a growth factor could elicit a signal in a cell. So we've got the growth factor that's bound to its receptor. This is a membrane receptor. It activates the receptor. The receptor then in turn will activate a phosphorylation cascade as part of its signal transduction pathway. And ultimately the last protein here will phosphorylate an inactive transcription factor. Activating the transcription factor will activate expression of a certain gene, creating the mRNA for that gene, which will then ultimately allow ribosomes to translate that gene into protein, and the protein can then function for the cell. Very likely, if this signaling molecule is a growth factor, then probably the protein that's made at the end of this transcription activation process, that protein will probably elicit the cell to go into cell division, for example, to go into mitosis, to allow for the growth of the tissue or growth of the organism. Ultimately then, once you activated a signal, you probably want to terminate it. If you don't turn things off within a cell, bad things can happen. This is one of the ways that cancer occurs. Quite frequently, you have mutations in those genes that help to control the cell cycle, and if the mutated versions of those proteins continuously signal the cells to go through mitosis, that'll overproduce that kind of cell, and that ultimately results in a tumor and a cancerous state. So it's bad to have signals constantly being sent to the cell over and over and over again when they are not necessary. And so an activation of these mechanisms is definitely an essential aspect of cell signaling. Typically, the way the cell or an organism will do this, it will essentially eliminate the signal. If the ligand concentration falls, then fewer receptors are activated because they're bound, and therefore fewer of the receptors will elicit an intracellular response. The unbound receptors will then also revert back to an inactive state, and they kind of just slumber there until maybe it is necessary to produce a signaling molecule again, and then once it's produced, the receptors can bind to it and activate the process once more. And then lastly, I want to give you an example of a process that is activated by cell signaling, and this process is called apoptosis. It's spelled apoptosis, but the first P is actually silent. It's pronounced the second P is silent. It's pronounced apoptosis. And apoptosis is essentially programmed cell death. So this is a way for a cell to identify through a signaling pathway that it's no longer needed for whatever reason, and it goes through a very controlled way of almost eliminating itself. Sometimes scientists will also call it cell suicide. And so during apoptosis, when the signaling pathway tells the cell, okay, it's time to go, the cell will essentially chop itself up and package itself into vesicles. And so these vesicles can then ultimately be cleaned up by scavenger cells that are part of the immune system. And this really prevents any enzymes from the inside of the cell just leaking out and potentially damaging other nearby cells. So it's a nice way to kind of recycle materials without creating damage to the greater tissue. Now, What kinds of circumstances would apoptosis happen under? Definitely if cells are damaged or infected, if they're not functioning the way that they're supposed to be functioning, the tissue will elicit the apoptotic response, and the cells will go through that programmed cell death. But that all is, it's very negative, right? You think that only cells are eliminated when things have gone wrong. But actually apoptosis is required as part of normal development, even in humans. So let's take a look at an example, right? When you look at your hands, most of us don't have webbed fingers. All of our fingers are separated nicely because what happened during development is all the cells that used to live in between our digits went through apoptosis. They were signaled to go through programmed cell death so that we could have individual digits on our hands and on our feet. So a very brief introduction to development. So when you are a tiny little embryo and the cells in that embryo are going through the process of differentiation, there are some cells that are going to create your limbs. And initially, these limbs are like little paddles. They're called limb buds. And literally, they look like little paddles. You can kind of see them here. There's really no separation between the digits. And once the limb buds reach that certain point in development, then apoptosis is triggered in those cells using a cell signaling pathway. In those cells in between the digits. And so in this particular figure, we're using fluorescence microscopy, and we're using a marker that glows a really bright yellow-green color in any cell that is apoptotic. And so you can see there's a high prevalence of those apoptotic cells that are glowing bright green in between what are going to eventually end up being the digits. And so this is a place where apoptosis is good, right? If it didn't happen during development, then we would end up with webbed fingers and webbed toes. But of course, also apoptosis can be involved in some diseases. So Parkinson's disease, Alzheimer's disease. In Alzheimer's disease, again, we haven't sorted out all of the pathology for the disease, but what we do know is that very frequently these improperly cleaved proteins will start to accumulate in the cells, and this prevents the neurons from properly functioning, and the cells are going to reach a point where they're so full of these misfolded proteins that they say, I can't do it anymore, and apoptosis will be stimulated through that signaling pathway, and the cells will go through programmed cell death. And this is why we have those cognitive impairments, memory loss, amongst other pathologies in Alzheimer's disease. And when you look at a patient that has had Alzheimer's disease through an autopsy, their brain tissue is significantly decreased. They literally physically have holes in their brain where the neurons, they've all just pulled the plug and they couldn't do it anymore. They've gone through apoptosis, and that individual has lost a lot of that brain tissue. And then, believe it or not, actually interference with apoptosis can contribute to some cancers. It's not super common, but if you imagine, instead of enhancing apoptosis, what if you prevented apoptosis from happening? That means that maybe some cells that should have been eliminated weren't. And that would result in technically an overabundance of cells. And that's essentially what cancer is, right? It can either be an overgrowth of cells or an underdeath of cells. If you don't eliminate them, then they exist in a space where they're not supposed to be, again, leading to a cancerous condition.